Great. Thank you very much. Good morning. My name is Ganesh Mochida. I'm a pediatric neurologist by training and a neurogenetics researcher from Boston Children's Hospital, also affiliated with Mass General Hospital. I work with Tim and his group on ASO development, also involved in natural history studies with Claudio and his colleagues, both in the US and in Brazil. And we have several friends and colleagues from Brazil, so I'd like to welcome them coming all the way to Boston for this meeting. And I also would like to thank Mary, Nina, Stephen, Izzy, and organizers and the entire AHC community for hosting this wonderful meeting. I feel that it's a great privilege to be able to work with each and every one of you on this endeavor, and I just cannot thank you enough. And so today I'm gonna briefly talk about the blood sample collection in EEG, which are done during the course of this conference. And I'm not actually representing our own study groups, but rather the entire research community, because as Nina alluded to, the idea is for the research community to share this invaluable resource. Um, so next slide, please. Yeah. So first about the blood samples that we intend to collect. So why it is important for your children to have these blood donated for research? The blood actually contains a lot of information. It has many different types of cells, many different types of molecules. And our idea is to find um, helpful molecules that helps diagnose, follow the trajectory of the disease, and measure the effects of potential therapeutic interventions. So to this end, we'd like to collect plasma serum and RNA, DNA, and in some cases, cells called peripheral blood mononuclear cells, which allow us to create induced pluripotent stem cells, which then in turn we can turn into neurons, Dr. Al George earlier mentioned. So from serum, plasma, and serum, we have many different molecules that we can measure. For example, we have metabolomics. So metabolomics is a sort of the entirety of small molecules called metabolites in the blood, starting from like blood sugar and amino acids and so forth. And we have next proteomics, which are much larger molecules, proteins. And then we also have uh, study ongoing for neurofilament light chain, which is known biomarkers for various neurological diseases. And we already have some preliminary data to suggest that it may be useful in monitoring the disorder in AHC as well. Um, there are also RNAs that Tim Yu earlier talked about. It's an intermediate molecule between DNA and protein, which we can detect from the blood and analyze comprehensively. So that's called transcriptomics. And then we have DNA. So many of your children had DNA testing to have the diagnosis of AHC, ATP1A3 related disorders, but we may be able to find other genetic variants that could potentially be a marker of certain phenotypes or potentially be target of genetic interventions outside this particular ATP1A3 mutation. And finally, these cells can be used to generate pluripotent stem cells, which are really critical in our ability to investigate what's really going on in the neuronal cells of the affected individuals. So we got together researchers and the organizations got together to come up with a certain sort of standard set of blood drawing. So we will try to collect the minimum amount of necessary, and then those will be either stored for future use or immediately applied to one of the research projects that some of them you've already heard about. In some cases, we may be asking you to have blood draw again in the future. Why is that? There are several potential reasons that we may be coming back to you and asking for future blood drawing. So one is that some of these measurements, it is important for us to have a trajectory. So not just a snapshot, but how it changed over time may be very critical. So maybe for that reason, we may need more than one time point. Also, obviously, as we learn more about the disease, we may have new ideas, which might require new or different types of samples. So that's another possibility. And finally, some of these samples do not last forever. For example, DNA, if it's purified very well, it lasts very long time. Probably you have heard about DNA studies from fossils and so forth. So it's very stable, but protein, not as much. So even with the ideal condition, it may not last forever. So for those reasons, it may be possible that we may request additional samples in the future. And these samples from the actual individuals really go hand in hand with animal models and other model systems that you heard from the researchers. So they are really critical in moving the field forward. So next, please. So EEG, I will not go into too much detail, but I just want to say that it's a non-invasive method of monitoring the brain activities. There are really two major part of information that we can get. One is the background activities, so that is also a reflection of a brain maturity, and then we can also detect abnormal electrical activities that are sometimes noted in children, particularly with those with seizure disorders. And we have several electrophysiology specialists, Dr. Arne van den Magdenburg and Dr. Christel Ashukar, Dr. Philip Pearl, and so forth, who will help in analysis of these EEGs. So I will end here. Just want to thank again for this wonderful community and including us in this great partnership. And thank you very much for what you do for your children, to each other, for the community, and your commitment to move the research forward. And thank you again, and enjoy the rest of the conference.